All right, welcome everybody to the webinar today. We are pleased to have Dr. James Westwood. He's a professor from Virginia Tech University. He has a Bachelor of Art in Biology at Concordia College in Minnesota. He has a master degree in plant physiology at University of Minnesota and a PhD on horticulture at Purdue University, Indiana. Dr. Westwood, thank you very much for accepting our invitation. We are very, very glad that you did, and we are looking forward to hear you.
Thank you very much. And I am happy to be part of this. I'm going to share my screen and bring up the presentation. And okay, you should see the presentation there. And so I want to talk to you today about plant-plant interactions, and specifically about the interactions of parasitic plants with their hosts. So, but first of all, when I'm introducing this subject to students in the US, I always have to give them an introduction to parasitic plants because we don't really see them or take notice of them much here. And I'm assuming it may be the same case in Brazil. So if you're not aware of parasitic plants, in fact, about 1% of plant species have taken this road to parasitism as a lifestyle. And so what you see in this slide here is just a sampling of the diverse species. And it is a fascinating diversity of plants. And what I'm showing you now in this more phylogenetic view is how you see parasitism represented throughout the plant kingdom. And if we you look in those family names that are in red, those are the families that have parasitic plant members. And I will talk about these ones highlighted in the yellow arrows here specifically today, but recognize that parasitism has evolved multiple times in different plant families. So it's really a time-honored way of life. You know, in all kingdoms, there are organisms that have found a way to steal their food from another organism rather than to make it themselves. And it just seems a little more surprising for plants, right? So when we think about the evolution of parasitism, these have all evolved from autotrophic plants. At one point in their history, they were photosynthetic, they had leaves and roots, but they somehow evolved to optimize this connection to another plant. And so they developed this structure we call the hostorium. And this is an organ that is unique to parasitic plants. It's only found in parasitic plants. And that's the connection, the bridge that connects them to the host plant, right? Then as a consequence of being able to take their resources from another plant, a lot of these parasitic lineages, they've lost the normal structures. They've lost the leaves, or at least fully developed leaves. They may not have a root system anymore. And certainly they've lost photosynthesis in many cases, with the ability to harvest their own light. There are many degrees of parasitism, though, so not all of them have gone completely to holoparasitism. But all of them, to one degree or another, have perfected or show some really exaggerated form of plant communication, because after all, to live parasitically, the plant must be able to locate another plant in order to attack it. And I will talk about that to some extent here. But the reason we study parasitic plants is, you know, in many cases because they're interesting, but, you know, they give us money because they're weeds, right? That's what we're about. We got to control plants and protect agriculture from weeds. And some of these parasitic species are among our worst weeds. I will discuss Striga, or the witchweed, today, and the Orobanche and Phelipanche, the Broomrapes, which are from the same plant family. And I'll also talk about Cuscuta. The other weedy parasite that I won't deal with is the mistletoes. And these are problems in the forest industry. And interesting also, but difficult to study. So to talk about the root parasites and the Cuscuta, just again, reasons why they're weedy. These can have a very tremendously high economic impact. So yield losses may be up to 100% when the parasite has basically re-engineered the relationship. So it's taking resources from the host. The host may or may not die. Usually it does not die, but the yield may all be diverted and may produce no seeds, right? So the yield losses can be up to 100%. If not that, the crop quality can be reduced substantially. Farmers who are faced with parasitic plant problems may have to switch to growing crops that are non-host crops, or even abandon the fields altogether in extreme situations. And the reason that some of these are so tough is because the parasite's often hidden from view. It's not above ground, or it's not readily visible until after the damage has been done. And control methods are indeed challenging and often prohibitively expensive. So to talk and introduce a couple of these more detail, I want to say something about the Striga, or the witchweed. Here it is, this beautiful purple flower, and it's attached to, in this case, a sorghum plant. It's attached underground. And so if you pull that up, here it is, here's the stem of the Striga along here. And this bit of root here is actually the host root that it's attached to. The Striga itself doesn't have much of a host root. It just has a host root system. It has that point of attachment and maybe a few other little adventitious roots, but not much. But the damage can be impressive with Striga, especially just at the point of that initial attachment. You can get a stunting of the crop. And so here's a field in Kenya. These should all be corn plants here. It's intercropped with a legume, so it's not just a terribly weedy field, although there's plenty of weeds in there too. But you see around each of these stunted corn plants, little cluster of the purple flowered Striga, right? So that shows you what it's done to what should be a healthy corn stand. And what's cruel about this plant is that it is attacking really your major subsistence crops, maize, sorghum, millet, rice, in a region of the world that is already suffering a lot of hardships economically, politically, just infrastructurally, right? So sub-Saharan Africa and down through Southern Africa, right? And so this is why it's probably, of all the parasitic plants, it's the one that gets the most attention and efforts to control it. But related to that are the Broomrapes. These are the Orobanche and Phelipanche species. They are the same family, Striga, so the Orobanchaceae family. The major difference is that these attack broadleaf crops, which can be very bad when you're attacking leguminous crops in parts of the world where that nutrition is important. But more of these are cash crops, sunflower and tomato and things like that. The other difference is that the Broomrapes are distributed primarily through North Africa, above the Sahara, and the Mediterranean region of the world. So they've kind of divided the world up in terms of both geographically and host crops. And so these are some of the major ones that we deal with. This is a picture of one of the Orobanches, Orobanche crenata on faba bean in Morocco. And you can see, there's a test trial plot, but you can see the beautiful flowers of the parasite here, the totally destroyed faba bean. Now let me show you the life cycle here. And this is just of one species. It's pretty representative, but there are, through this life cycle, there are several points of communication with the host plant. And the first one I'll spend some time talking about, is the germination stimulant. This is kind of maybe the most famous example of the host-parasite communication. So that seed of the parasite sits in the soil and will not germinate until it detects the presence of a host root nearby. And I'll go into that. But once it germinates, it produces a tiny little radical. That radical then uses a second chemical signal to initiate the development of the hostorium. The hostorium then will form attachment to the host root here, and then you get the integration of growth, right? You've got parasite cells growing in complete contact with host cells. They've got to invade that host tissue and make vascular connections. So there has to be a coordination of growth at some level during that stage. And then, kind of as a continuation of that, the parasite has to suppress host defenses and convince that host plant to reallocate resources to the parasite. So at this level of signaling is where I've spent a lot of my career, and I think until recently has not gotten much attention, but it's getting, there's a lot of good things going on now in this area. Ultimately, this parasite develops into a bulbous structure, produces a shoot, and appears above ground. And that's the only visible part of this parasite, is the flower. And then it continues the life cycle. So let's go back and kind of review the major steps here, looking at a parasitic plant almost as you would a pathogen, right? So challenge number one is to locate an appropriate host. And I'll talk about Strigolactone signaling and other mechanisms briefly, and then move on to talk about not so much about attachment, but about these processes that go on when you have the parasite growing within the host, intercellular growth, the extraction of water nutrients, and the suppression of host defenses. And that'll be the kind of the second part of the talk. But right now, let's focus on host location. Now, I told you that these seeds were small. Here are some Striga seeds. Of course, here they are very enlarged, but in fact, they are extremely small. So here they are, this is human hair. And this picture over here, the very small particles you see in this picture are the seeds of the Striga. So if you're holding it in your hand like that, it doesn't look like a plant seed at all. It looks like you're holding, your very fine coffee grounds, maybe. But these fine seeds need to do a big job, right? They need to make a germination decision, right? And this is critical because these seeds are extremely small, as I said, limited reserves. They can only grow a radical that is maybe 1 to 2 millimeters long. And so they've got maybe a week after germination to make contact with the host and make a connection and grow. If they don't do that, they're dead. So it's suicidal germination. There's been quite a bit of effort put into the idea of trying to get seeds to spontaneously germinate without a host around and undergo suicidal germination. This is the weed scientist dream, right? To control the seed bank. Theoretically possible, but in practice, extremely difficult. So what do we know about this signaling process, right? We want to talk about the communication. So it has been known for really nearly 200 years that the hosts were producing a chemical signal that was triggering germination of Striga. And the first compound, Strigol, that stimulated germination was isolated in 1966. And still a long time ago. Right, but there's been a lot of work in recent years that has made this into a very nice story. Basically, here is a Strigolactone molecule over here. You see it's got 3 rings with a 4th ring hanging off over on the end here, the D ring. This is exuded from the roots of the host plant, and the parasite detects that. Now, that seems simple, but it gets more complicated. We thought there was one stimulant, maybe one receptor, but I'll tell you that is not the case. But here's kind of the dream of the breeders and the parasitic plant scientists, right? Can we solve the problem using this host stimulant specificity? And this is a picture from a recent paper. There is susceptible sorghum growing over on this side. You can see the pretty Striga flowers there attacking that plant. Whereas on this side, the sorghum, it's a stunted American variety, well, a dwarf, I should call it stunted, but it is producing low amounts of strigolactone, or in fact, really a different type of strigolactone. And therein kind of lies some of the promise of this method. So one of the questions that stood for many years was, why would a plant produce a chemical, a strigolactone, that would reveal its presence to a parasitic plant nearby? Well, it turns out that strigolactones actually are plant hormones, that they have very important functions within plants and with the organisms in the surrounding soil. So a big discovery was that strigolactones are exuded from the host, from the plant root, as a signal to our arbuscular mycorrhizal fungi. And this is an ancient symbiosis, right, whereby the fungi associate with the plant in this beneficial relationship where the plant actually uses the mycorrhizal fungi to gain water and nutrients that it wouldn't ordinarily be able to gain. So that's an important component to plant fitness. At the same time, these strigolactones are circulated throughout the plant like a good plant hormone. They move upward in the plant, and they play a role in controlling shoot architecture and many other processes that I won't go into. But it tells you why it is hard to just knock out the synthesis of strigolactones in a plant and still have the crop you want at the end of the day, right? There's a price to pay for disrupting this pathway completely. And through evolution, the parasitic plants have learned, well, adapted to pay attention to these compounds, right? And that's how they germinate. So a little more about strigolactones. So turns out there's not just one, there are several. In fact, there are many. And I just want to show you again the structure here. It turns out that this D ring, which looks identical in all of these images, is very highly conserved, and that's an important part of the compound. The rest of the molecule can suffer all sorts of changes and modifications, and that will affect specificity, but it may not affect the D ring is really important there for the activity. And now here, I show you full, more complete families of strigolactones. So you can see the breadth of diversity here, and I won't go into this very much, but we break them down into a couple different types. There's the Strigol type and the Orobanchaceae type strigolactones. And these are separated really based just on the orientation difference between the B and the C rings, right? Just how that molecule is folded has an effect on how it is perceived by the plant and the parasites. So there's a lot of good stereochemistry here. If you like your organic chemistry, there's always good questions for chemists to solve. Now, the other side of the question, you've got plants that produce a range of strigolactones. We don't really know why, but each plant species will produce its own subset of all the diverse strigolactones. And it's not clear what is pushing this diversity, whether it's probably not driven by pressure from parasitic plants. I can't imagine parasitic plants are shaping strigolactone evolution that way, but there must be other reasons. So that's a good open question. And we know that strigolactones are generally perceived by a receptor here, and it's called D14. And it's cartooned out here to bind to the strigolactone. You see that the D ring there is cleaved off, and once that is cleaved, the shape of the D14 changes in such a way that it can associate with another protein. This one I'll just call SMAXL. It's one of these genes that is a transcription regulator, and it actually sits on DNA on a gene regulatory region and inhibits the expression of that gene, so that when they get together, D14 with the SMAXL, it leads to the degradation of the SMAXL inhibitor, and the whatever gene it's suppressing is now active, right? You can see how this could lead to then a nice change in plant phenotype, whichever gene is being regulated. This is, in fact, a type of hormone system, or hormone recognition system, that is also seen in things like jasmonic acid and the auxin hormones. So it's a similar type mechanism. Now, that's how plants perceive their own strigolactones. What about the parasites? So they actually are using another gene called KAI2, or keratin insensitive 2, which turns out to be a very close relative of D14, a close homologue. And if we look at this in a kind of a family way, you can see that D14 is here. Its ligand, the strigolactone, is shown here. Next closest related receptors, the KAI2, which seems to have evolved to detect either keratin or something like it. Now, keratin is a compound found in smoke. This is a great ecological story. When you have a fire, there are certain species that will germinate after the fire, and those are responding to keratin that's present in the smoke. And so there's an ecological value in that. Most plants have a KAI2, so they're probably responding to something besides just smoke. There's got to be something else in plants. But this family of KAI2 genes has expanded in parasitic plant, in the Orobanchaceae parasitic plant lineage, to include some other versions. There's the conserved version, which is KAI2C. We don't quite know what that's doing. Most plants have that. But then there's the intermediate and divergent form, which are really special to the parasitic plants. And these are the ones that seem to have evolved, kind of re-engineered to recognize strigolactones again. All right, so this is what that phylogeny looks like in the real world. The D14s are here, 1 copy per plant. KAI2Cs, 1 copy per plant. Same with and the eyes, kind of hit or miss whether a plant has them. But the KAI2D genes, represented among several different parasitic plants that have been sequenced, is a diverse range. So why? Well, we think that the parasitic plants have evolved a kind of a whole team of KAI2D receptors that will recognize different flavors of strigolactone produced by different hosts, maybe. This actually hasn't quite been proven. yet, but it's a good working hypothesis. We still actually don't know if all of those KAIZd-the genes have a function or not. But what I want to end with for this part of the story is a kind of a summary of a situation that seemed so simple about 15 years ago, and it's now really rich and complex. You've got Strigolactones, a whole bunch of them being produced by basically all green plants are producing some set of Strigolactones, and they have important functions both within the plant and exuded into the environment to communicate with mycorrhizal fungi and maybe other organisms. But that has been seized upon by the parasitic plants, who have evolved a new set of genes just for recognizing those and germinating with precision to make sure that they are germinating only when their preferred host is nearby. So a great story of plant conversations going on, and certainly more to be discovered about that. So with that, I want to move on to, I want to talk about a different parasite. And this is Cuscuta, or dodder. It's a stem parasite with a wide host range. And I want to show you my video here. So what you will see here, I'm going to see if I can silence my speakers. If you can still hear me, what you notice is the dodder germinating here in the soil. And there's no special germination cue for this parasite. No, it's just going to germinate in the spring when normal plants are germinating. Here it is, though. It has to find the tomato plant, and it's reaching over to the tomato plant. And you notice it seems to know where the tomato is. It's not leaning in the other direction. So by some combination of cues, whether it is volatile signals or color, light cues, I think probably, but there's evidence for these. And so it finds it, and it makes a physical contact, and then it wraps around. And where it is wrapped around the host, it forms these bulges, and each of the bulges is the site of a hostorial formation. And those hostoria grow into the plant and make vascular connections. So if you try to rip the Cuscuta away from the host plant at this point, if you leave the hostoria behind, it can form new shoots from the hostoria. And so you get this proliferation of shoots. And as the plant is growing, it is constantly branching, with each new branch able to form new attachments. And so what you see there is the new shoots searching and touching the plant, and where they make the right connections, they will wrap it around and continue to grow like that until really they've covered the whole patch. And with that, I will move on, and I will tell you a little bit about what you were looking at there in terms of the hostoria. So here is my cartoon of the Cuscuta growing on in Arabidopsis here. And if you make a cross section through this is where the hostoria are growing into the parasite. You see something that looks like this, right? Where the hostoria are making connections with vascular bundles, and they make at least 3 types of connections. They grow within and between the cells. They make the vascular connection to the xylem. They make contact with the phloem cells, and no open pores that anybody has demonstrated, but there is transfer cell activity. So it's definitely transfer going on there. There's also these cases where the parasite cell has grown through a host cell and laid down a host cell on one side, parasite cell wall on the other side, with plasmodesmata that span these. So you have this incredibly close connection where the host and the parasite have potential to exchange cytoplasm, right, with each other. And so really close connections. If you imagine this also with parasitic plants forming connections between hosts. So Cuscuta is one of the few parasites that will actually parasitize multiple hosts at the same time. Here it is on a patch of lentils, and here it is connecting 4 different species of plant together in a single plant. And just appreciate that these networks can exist quite in very dense ways. This is in cranberry, actually in the Northeast US. This is a cranberry bog, all connected by parasitic plants, and all by Cuscuta. So at this point, I want to talk about Cuscuta in specific and these types of connections, the intercellular growth, extraction of water, and the evasion and nutrients, and the evasion of host defenses. And so we think about the things that are being exchanged and what it really means. So they all exchange water, we know that. They exchange sugar and small molecules. Yeah, that's what the parasite is after. And in this classic figure, you can see a host is on the bottom, the parasite is up here, and this dye, flow-mobile carboxyfluorescein dye, is showing up green here. You can see it moving across that hostorial bridge there. So that's a small molecule. Work from my lab has shown that there's also the exchange of messenger RNAs. And just to orient you to what you're going to be looking at in several experiments here, here's a stem of a host. In this case, it's Arabidopsis with Cuscuta wrapped around it. And all we did was take tissue from the parasite stem out here, the host stem up here, and then the interface region, which is a combination of both tissues, and did deep sequencing, right? Next generation sequencing. And we were able to basically reconstruct, here's a host gene, able to reconstruct all those reads just based purely off the parasite stem. So we were able to show that the messenger RNA was moving in that direction. In fact, it's moving in both directions. And so here's just some gene counts. And when you look at the number of different genes represented in those sequencing libraries, we found thousands of Arabidopsis genes represented in the RNAs in the Cuscuta, thousands. Well, without a good Cuscuta genome at the time, these are unigenes, so it's probably less than the number there, but still in the thousands of genes represented from Cuscuta in the host. So, well, what's going on? Well, we know that RNAs make proteins. So do proteins move? And well, yes, there's actually some old literature using the green fluorescence protein in a way that was just like that carboxyfluorescein experiment I showed you. Here's the GFP moving up from the host into the Cuscuta. But there's a size limit on that. And our question has been, will a mobile RNA be translated into a protein in the destination species? And so we've done an experiment here where we grew Cuscuta on, in this case, a plant, a Arabidopsis plant expressing the Gus reporter gene. This is a Gus reporter gene, produces a blue color when you provide it with a proper substrate. In this plant, the Gus has been fused to a tRNA motif, which confers mobility on the RNA. And then we grow Cuscuta on these, and we cross-section, and we stain for Gus activity. And this is what we get. Again, here's the setup, cut like this. Normally, the host it's round in cross-section here in the Cuscuta, here with the asterisks marking the hostoria. So if you look at the plant with just the Gus construct. Here is the Cuscuta. Out here, we don't see any sign of blue, although the host is very blue because it's, of course, expressing the Gus gene under this strong kind of universal 35S promoter. When we look at the Gus fused to the mobility element, we see blue color out here in the parasite. And so to us, this tells us that the RNA, once we made it mobile, is now producing a color out here. So we assume it's being translated into the Gus enzyme in the parasite cells. Okay, so this got us to thinking about whether we could see actual proteins from the host in the parasite. And so instead of doing the experiment with searching for RNAs, here we're using LC mass spec to look for protein signatures. And in fact, we found in the parasite 447 Arabidopsis proteins, and over in the Cuscuta, nearly 100 Cuscuta proteins, based on what we could confidently identify. And of these, again, they're moving both ways. In the Arabidopsis, of those mobile proteins, 271, about 2/3 of them. We also had evidence from our data that they have mobile RNAs, kind of a similar proportion from the Cuscuta. So we can't explain all of them based on our data, but a certain number of them could be explained by mobile RNAs. But that alone is hardly convincing. But if you look at the data here in terms of its size distribution, and so what you're seeing here is Cuscuta. In the purple, the Cuscuta proteins that we found in Arabidopsis, so there's fewer of them than the Arabidopsis proteins we found in Cuscuta, broken down by size. So the small group that has most of the proteins, yeah, they tended to be small in the 100 to 500 amino acid range. And this is about the size of the green fluorescent protein, which I've showed you is already has mobility. But up in this range, we're talking about, this is the Gus protein, 600 amino acids, not usually thought about as a mobile protein. And here again, we're seeing quite a few representatives of a foreign protein in either the host or the parasite. And even in a few cases, we found evidence of just huge proteins, over 2,000 amino acids. And this was only in the direction of the Arabidopsis to Cuscuta, but some evidence there that there could be massive proteins. And the easiest way for me to explain this is, again, through the transmission of a linear mobile RNA that is subsequently translated in the host or in the parasite. The other way to think about this is in terms of water solubility. So among those mobile proteins, we saw some of them are water soluble. Yes, they would be expected to be flowing through the phloem or the cytoplasm perhaps. But a certain percentage of them, in this case, about 20% of each of our data sets, are either membrane integral or membrane bound. So they should not be moving through any translocation stream. And yet we find them showing up in, from Cuscuta to Arabidopsis or vice versa. And these things are harder to explain by mobile protein. And so I think we are seeing some evidence that RNAs are moving and they are translated. But it still doesn't tell us whether it means anything. And so I'll just share one slide here about the work of a previous postdoc, Soyan Park, who was characterizing this gene called GYP23. And in our early protein experiments, we found this as an abundant protein in Arabidopsis parasitized by Cuscuta. It's a small protein, very poorly characterized, but it was reported from a jasmonic acid experiment called jasmonate-induced protein of 23 kilodaltons. So we call it GYP23. And this is interesting because, so it shows up in our data. So it's from the parasite, it's high, of course, it's made in the parasite. There's no corresponding gene in Arabidopsis. And yet it's in the interface and at low levels in the host stem. So the RNA is mobile, and the protein is mobile. So both of those are mobile. We don't know which one is moving or not, but somehow the protein is getting into the host. And through some experiments, yeast 2 hybrid, she found that it is interacting with a protein called KAT2, which is part of the JA synthetic pathway. And so our current hypothesis is that this GYP23 is a protein that is being made in the parasite, whether by RNA or by protein moving into the host and influencing the regulation of the JA pathway. And JA, as you may know, is one of the kind of common defense pathways in plants. And so it would make sense that this is going on. In addition to this, we've got one more example of RNAs, and that's the microRNAs. And just to remind you, RNA has numerous functions. So messenger RNAs, as I just told you about, are encoding proteins. MicroRNAs make small, little, about 20 to 24 nucleotide long RNAs that can find homologous regions of a messenger RNA, bind them, and actually inhibit protein production. So they in fact silence certain genes, right? And so by this way, small RNAs, microRNAs, have a regulatory function. And so this experiment, I will just briefly tell you, is in collaboration with Mike Axtell at Penn State. Basically, we did the same thing as we did for the other RNAs and protein, and we found that there was an enrichment of microRNAs in the interface region. And so here, not so much in the stems like long distance trafficking, but more where there's the real integration of the host and the parasite. And what you see here in this blot, you see the blot, you see that's a high level of these small RNAs that are targeting some specific host genes. And I'll just show them here without belaboring them. BIC1 is known in plant pathogen response. SOR1 is probably the most interesting. It's a sieve element occlusion factor, so for plugging up the phloem, and some auxin-related binding proteins. All of these, well, these few have been found in other experiments with pathogens trying to shut them down. The sieve element one is especially interesting because Cuscuta is a phloem-feeding parasite, right? And this experiment is just showing you that we see a suppression in the level of gene expression that represented by the red data for each of these target genes. This is the control over here, relative to the control, which is in blue. So we actually see in the interface region the suppression of the target of the parasite microRNA. And then if you get an Arabidopsis mutant of that same target, so here's an Arabidopsis plant producing less of the SOR1 protein, it should be more susceptible because it's actually deficient in the thing the parasite is trying to shut down. And in fact, in this experiment, it was. Here's the control wild type. Here's the SOR1 mutant, and we're measuring Cuscuta growth, so it's growing better, significantly better here, and also on one of those auxin binding mutants. So to summarize here, what I've showed you is that there's a lot of things being exchanged. So here's my host cell and the parasite cell diagrammatically represented. And they're both producing messenger RNA, and they're exchanging some of these RNAs. And they're also making protein and exchanging that protein. And at some level, perhaps they are actually translating protein or are translating into protein from messenger RNAs that have come from the recipient, another organism, right? Now, in addition, the parasite's making microRNA, which is moving into the host and shutting down gene expression there. And the last part is, I didn't tell you about this. This is previous work by other people using host-induced gene silencing to shut down parasite genes by a similar mechanism to the microRNAs. So there is a lot of exchange going on here. And what we propose is that we are adding RNA to this kind of already existing suite of communication between the parasites and their hosts, as when viewed as like plant pathogens. So here's the parasite and the host, and they're exchanging factors that, as I've discussed, chemical signals or proteins that may influence recognition and defense response. And we think that the RNAs and certainly proteins are all part of this conversation and part of how the parasites are doing their jobs. So with that, I want to conclude, and hopefully the main messages you've gotten out of this talk are that some of the world's worst weeds are parasitic plants, and that parasitic plants are exceptionally good at communicating with their hosts through on multiple levels, multiple different ways of doing things, and through the microRNAs, messenger RNAs, proteins can all be used by Cuscuta in ways to manipulate its hosts. And I might have gone a little bit over my 20 minutes there. I want to acknowledge my collaborators, who I have many of, and funding agencies. NSF has been very good to my program over the years. And I will leave it at that and take some questions, Luis.
All right. Thank you, James. We are open for questions. We have a question from Dr. Cássio Mendes. Congratulations, Dr. Westwood. Does the addition of biochar in the soil sorb Strigolactones and inhibit the germination of the roots of the parasitic weeds? Do you know?
Oh, so I'm not sure if I can comment on biochar. I have seen some experiments that I'm recalling from years past where they used something like that, a charcoal or something absorbent, and the effect was yes. I think you can, if you can absorb up those compounds, it helps make the host invisible to the parasite. I think in a practical nature, it's harder to do that. But organic matter will work. If you can increase the organic matter of the soil, you can shift some of that communication, I think.
Mm-hmm. You can ask the questions on the question and answer session. I have a question, Dr. Westwood. do you know any effect of the climate change, like CO2 or any other environmental stress that is affecting the spread of the parasitic weeds? Do you know anything about that?
I do not know specifically about climate change affecting this. I do know that parasitic plants are spreading. Whether that's all due to climate change or just due to human activity, I can't say. I know it's spreading up into Northern Europe. England is getting a problem with some of these parasitic plants. Increasingly, they're moving to other countries. Again, probably as much due to man-made movement of things as to climate change. Climate change can't help. I'll just say that.
Yeah, that's true. And regarding you have been showing in your talk about the trying to control the host parasitic weed interaction by resistant plants. Do you expect any like weed resistance to this resistant plant in the future?
Yeah, I do. Yeah, I mean that's a great setup. I think we've struggled so hard to find resistant host germplasm, and they're making some good progress in some research groups. But what I showed you about these parasitic plants and their ability to adapt, all those receptors for different strigolactones. I think these are really innovative plant species, and the selection pressure is super strong. So for them to live, you will find plants that can overcome resistance. The breeders will have to work very hard and very fast.
Yeah, that's true, because the weeds adapt.
They're so good at it, and there's so many of them, right? The genetics are against us.
Yeah. Andrea has a question here. Thank you for your interesting lecture. Recently, a team from the University of Pelotas published a study where it was observed that the interaction from a distance of the host with the Cuscuta changes the electrical signaling of the Cuscuta. Do you think studies on parasitic and electrophysiology have some potential for fighting or achieving important breakthrough parasitics in crops?
Great question. I had some high school students over the summer, and we tried an experiment with electricity because we've seen it coiling around electric cords in our growth room. I have to go find this paper you mentioned. I would not at all be surprised if there is an effect of electrical fields or magnetism or something like that. Very plausible to me. We were not able to prove anything with some simple experiments. We could not find anything conclusive in terms of attaching to wires with current or not. Yeah, what a cool idea.
Yeah. It's impressive how the plants manipulate the host. It's amazing.
Yeah.
And in the small interference RNA are some unique findings, because the weed make the plants to produce the protein that it needs to be interactive with the host. It's amazing.
Yeah.
Let's see if we have any other questions here. I think we don't have any questions. Okay. Dr. Westwood, it was a very, very interesting talk. We learned a lot today, and I can see that the evolution of the understanding of the plant-host interaction is moving quite fast. And we know that your team is doing a very good work on help us to understand that, and we appreciate your sharing this information with us.
Thank you. It's very nice to talk to you.
All right. Thank you very much.
Right.
hope to see you next year.
Yes, I will. Yeah, we'll definitely. I'd like to see you in person when we get back to that.
All right.
Yeah.
Good. Yeah. Bye-bye.
All right. Bye, everyone.